All right. Hello, all you invertebrate enthusiasts. Pardon me, clearing my throat right at the start. This is gastropods that I love and that I wish that you know about and love too. We'll call it Lecture 23. Remember, with the gastropods, we're talking about the conchiferans, which includes the monoplacophorans, the cephalopods, scaphopods, bivalves, and the gastropods. This is the group that we're dealing with until the end of next week. Here it is here in the more complete clade that we introduced to you on Wednesday that includes the monoplacophorans based on some samples that were collected back in 2008. And remember, when we're talking about the gastropods, we're talking about enormous modifications of the shell, the mantle, and the foot. In this case, we're going to spend some time today talking about the shell. Some other adaptations in the foot as well, but we're going to start out talking about the shell. The shell, which could look like this kind of valved polyplacophoran, a single shell, a single kind of a plate of a monoplacophoran, or the cona and plana spirally coiled gastropod. And we're going to start out talking about some adaptations, incredible adaptations of gastropods found relatively recently, just at the very end of the last century, in 1999, at a steaming hot vent, deep abyssal, just off the coast of India. And this was a case of a snail that uses iron sulfide. Stop. That uses, Doug is here as well. And he's clearly excited about this hot vent gastropod that uses iron sulfide both in dermal sclerites on its foot and also within the shell. This is kind of a never before seen adaptation. The scaly foot gastropod, so Chrysomalon squamiferum. And you're looking at a video here that is taken of a couple of, several of them that have been sampled. And these scales on the foot that you can see there, the scaly-footed gastropod, contains sulfides and other metals. And it reinforces the idea that many bioengineers and biomimetic scientists have had that fortifying biominerals that mollusks use essentially reflect the availability of the minerals in the environment. So if it is there, they will find a way to use it. Here's a couple of looks. So we've got the scales and then in two different sections here. So this is after preservation in ethanol. So the golden appearance is an artifact of preservation. They would appear black. And what we've got here is two cross sections. One in just from a microscopic view of the sulfide, the black sulfide sulfur that's covering up the scales containing the protein and the conchiolin, and then the pulp conchiolin and sulfide metallic kind of coating covered in an SEM. Now, this is not just true for the scaly foot. This is actually a recent discovery that it's actually incorporated that they use this ferric material, this iron, in the shell as well. And so the familiar layer of the calcified layer, the organic peristracum, is covered in this case, the proteinaceous layer is covered in iron sulfide. And so you can see here in a non-scaly foot snail what that ought to look like. And it's a very different appearance. Now, we talked about this a little bit in the on Wednesday about the formation of the shell and that it's laid down on this outer lip, and the coiling, the plana spiral coiling, ends up being caused by the fact that the outer lip is growing faster than the inner lip. So you can see the mantle layer here, the outer lip of the mantle is producing faster, and so that ends up kind of passing on the outside. Now these are laid down, and there was a question in the chat on Wednesday about whether or not these are laid down as a left-handed or a right-handed manner. And in fact, most snails, maybe as many of 90% of them, are right-handed. Most species are right-handed. In some species, they can have both left-handed or right-handed. So the left-handed shell, if you put your cursor over the bottom here, you can see the left-handed shell is coiling to the left, and the right-handed shell is coiling and opening then to the right. Now this is a problem, and this maybe you saw this in the news. If there is a mutation within one of the species that is predominantly right-handed, you end up with a left-handed shell, where in this case they call Jeremy, even though they're hermaphrodites, where they were unable to mate with each other because they were a left-handed snail in a right-handed snail species. So you've seen this. This is talking about the mantle and the shell before. So the epidermis of the mantle laying down the nacris, the prismatic, and the periostracum, that proteinaceous layer. Now we didn't talk as much yet about the actual. We talked about the mantle being three-lobed, but that's kind of an important adaptation here, and you can see that the outer mantle, the outer lobe, this is what secretes. The inner muscular lobe, this is what tends to be more muscular, and then if there's any sensory adaptations, photosensory, chemosensory, we'll see both. Eyes in the mantle. We've seen eyes in the shells, and now we'll talk about seeing eyes in the mantle. But you can see the three lobes of that mantle essentially provide three different surfaces where evolution can kind of modify that body plan. So one of those modifications involves laying down from that third mantle, from the outer lobe, which secretes the cell, limpets. And we've seen limpets already in the news because the hardest teeth in the hardest natural substance in the world, we learned, were radular teeth. We did this on Monday, of the limpet. A different species of limpet is called a blue-rayed limpet. And it contains these prismatic layers that are laid down in these gorgeous fashions, almost like cat's eyes that you would see in a highway at night. And this is a highly conspicuous, mineralized, photonic layer that is achieved by this limpet shell. And we've got a video to show you what it looks like here from the supplementary materials of that Nature Communications paper. But you can see, based on the angle at which you approach, with very little light, you see these highly conspicuous layers of blue, apparent blue. Now this is caused by the fact that in the periostracum and the ostracum, there are these periodically layered zigzag architecture of calcite lamella. And these occur beneath this photonic multilayer that have colloidal properties. And that disordered array of light-absorbing particles provides contrast for the blue color and ends up producing these blue stripes in the limpet. Now, what you might not know about limpets that have this conspicuous blue coloration is that they can't see it. So this is not coloration that's for intraspecific communication. And what the authors hypothesized in this great paper in 2015 was that the coloration in the limpet shell has actually evolved, potentially as an example of Batesian mimicry. That if you look at the distribution of the limpet here in blue in Northern Europe and all the way down to Western Europe, Western and Southern, on the coast, that their range overlaps with a lot of these very visually blue and distasteful nudibranchs, other snails. And their hypothesis is that this striping pattern might serve to mimic toxic or otherwise distasteful organisms, even though the limpet themselves is not. So this is not a case of the limpets speaking to other limpets with the blue indicating health or anything like that. It seems to be a case where the limpets are speaking to predators going, you probably don't want to eat me. I might be as toxic as that other nudibranch that I share a distribution with. That's pretty amazing. Engineers are using this technology to try and figure out how to generate better, actually, cat's eyes in highways. So the creep sole, one of the other synapomorphies for the gastropods is, of course, this is gastropod, is that foot. And there's a number of different ways that they move. The pattern of movement within the foot can be directly monotaxic. It's kind of waves moving forward. There can be retrograde, where waves of the creep sole move backwards. There can be didactics, where there's actually separate but parallel contractions that are seen. Now, what are these actually? This is kind of a useful diagram, perhaps, maybe after you've seen what these waves of the creep sole actually look like. Now, if you've got snails in aquarium at home, you've seen this before, but you can see these anteriorly proceeding waves of the ciliated creep sole that, when lubricated with mucus, provide traction and locomotion for the gastropod. Gastropod mucus we've seen already in terms of that stylet that allows it to eat. Mucus is really important to the gastropods. It provides locomotion like we just saw. It can provide some case navigation. It can provide a homing trail. They can retrace their steps. Predatory defense, water conservation, and estivation or dormancy. Now, one example for the dormancy that mucus is really important for, associated with the foot, we can see in this snail, Sepia nemoralis. This one is in the dairy bush on our University of Guelph campus. And Sepia nemoralis is a snail, a terrestrial snail that does go through long periods of estivation. This is one particular study that looked at tree climbing behaviors associated with Sepia nemoralis, associated with the distance climbed and the relationship between the how much humidity there was, how much rain. So for the snails in our dairy bush, it can occur at any time. It's not a seasonal thing. It's really a direct response to water. And when that snail estivates, it retracts into the cell and secretes a mucus veil around, and by which they attach themselves to a plant or a tree trunk, and that seals off and keeps a moist interior as the dry exterior might be going through a long drought period. So perhaps a condition that some of these terrestrial snails might end up using more and more is in an anthropogenically forced climate change kind of a future. Now, snails, one other neat observation or kind of evolutionary story about snails is, as we said, hermaphrodites, but one of the ways that they inspire one individual to act as a male or another as a female is through the use of what Richard Chase at McGill University called love darts. So when two snails are intertwined in mating, one snail shoots this love dart into the body of the other. That rather drastic form of courtship is the dart shooter is then stimulated to produce sperm, whereas the dart receiver appears relatively unmodified, and so then begins to act as and foster that sperm and plays the role of the female. The bottom picture, you can see lots of SEMs of different kind of darts. They're gorgeous kind of structures that taxonomists like because it provides lots of different characters and character states that help differentiate one species from the other. And if you're wondering where the dart is in action, there it is, kind of piercing the foot and the head of that snail. So donor, recipient. Snails, of course, exist across the world in all sorts, practically every habitat that you could think of: marine, freshwater, terrestrial, benthic. They burrow, they can be pelagic, they can be sessile, they can be interstitial. There's snails that feed on hydrozoan like cnidarian jelly, then secrete chitinous bubbles from which they hang and float pelagically in the water. There's a glaucus that feeds on manowar and sequesters the nematocysts of those cnidarians to use on their own. Ancestrally, of course, we imagine them to be herbivores with that paint-scraping radula, scraping paint or scraping plant material and algae. But of course, they can be carnivorous as well. Many of you will be familiar with this cone snail that has lost the odontophore and modified the radula such that it's basically a small harpoon. There's a great video segment that the National Geographic has put up online that I'll jump into the middle of. You can go and take some time later on if you wish to watch the whole video, but let's take a quick look. So they can also be raptorial feeders, moving through the water column. They can be suspension feeders. Nudibranchs, which you heard about from Dr. Layton, can graze on cnidarians and save, as we talked about, and use discharged or undischarged nematocysts. Sea butterflies prey on shelled sea butterflies, and where the shell-bearing sea butterflies are suspension feeders that trap particles in their mucus, so suspension feeders, and then I'll show you a video of that in a second. But the other is that these mobile forms, there's incredibly gorgeous examples of convergent evolution where these mobile shellless forms have convergently evolved essentially forms that look and swim like a fish. You can see here in the picture, and there's what one looks like. So swimming, as you can imagine, a small rock bass even swimming in a terrestrial Ontario kind of a sense. There are other marine shellless forms, maybe with a long proboscis here looking almost like the head of an elephant, but this could be, if you think about slugs as being the only example of shellless gastropods, that's not in fact correct. There's many more species of marine gastropods that have secondarily lost their shell. The raptorial feeders
beneath the breaking sea ice,
are talked about here.
A predatory sea slug flies through the water
so this is the predator.
Wings. It's on the trail of
and this is the suspension feeder.
Snail.
So both living pelagically in the Arctic under the sea ice. They also can be deposited in scavenger feeders. This may put you off snails. This is a small rotting fish dropped into a tiny bit of sand in a benthic zone, close to a beach. And this is the eastern mud snail appearing like zombies in The Walking Dead to take care of that rotting fish. It goes on. Puts me off snails a little bit. But this eastern mud snail feeds on detritus, diatoms, algae, dead plant, and you can bring it out in low tide zones by finding or laying out little bits of decaying fish material. There's another amazing, one of the last examples that I'll talk about today in terms of what they eat. Well, some snails, so suspension feeders, herbivores, carnivores, raptivorous, sea angels, and we have slugs, sea slugs, snails, that are potentially photoautotrophs. This is kind of crazy. This is called Kleptoplasty. This is a particular species of slug called Elysia, it's a genus name, where the genes that support photosynthesis have been acquired by the snail by a horizontal gene transfer, and the encoded proteins are then targeted to the plastid. Wait a second, plastid you say? Well, in case there's the grazing, so this ancestral condition of grazing on algae, this appears to be one case. Another example, like we saw in protozoans, as we talked about the serial endosymbiotic theory of how complex eukaryotic life came about. But a case where a chloroplast was partially phagocytized by a digestive gland cell, but not completely. And the chloroplast, which you can see here, continued to grow. And it was living. And so the green that you're looking at here in the Elysia, these are actively photosynthesizing chloroplasts. And as I said earlier, that chloroplast, this relationship has been going on long enough that all of the genes to complete photosynthesis are no longer contained within the plastid of many of the algal cells that this snail eats. The nuclear gene, they've transferred over to the snail genome. So this is a complicated chimera of a plant animal, an animal plant. Now, this is kind of incredible, and it hasn't just happened once, it's happened multiple times. 3 nominal taxa, 20 different candidate species where we've seen these kinds of nidoplasty, and these are super productive areas to think about for drug discovery, plastid symbiosis, biological control, a whole bunch of things. And in fact, this species has been in the news as of this week because a study that came out on Monday that looked at this species talks about the fact that it has an extreme, never before seen form of autonomy. So you can cut the head off, and they can do this to themselves. Remove their head from their body. The body will die. It doesn't grow a new head. Within that body, that's the visceral mass. So you've got the gut, most of the lungs, everything. You've just got the head at the anterior end, but you can see it's still moving. It's crawling around. It will regrow the rest of that visceral mass. It doesn't have a gut to do it. And one of the reasons that they think that the authors hypothesize that this kind of extreme, incredible form of autonomy is possible is because there are plastids here, and so essentially it can grow as a plant until it regrows its animal stomach. Why would you want to do this? They further hypothesize that this might be a way, if your body is too full of parasites, you cut it off, you take your head, and you regrow a new body that's parasite-free. That's kind of amazing. Like, kind of amazing. So there's clearly going to be more work about this that maybe you can participate in the future. Gastropods themselves are eaten by a lot of things. This is a video that I took in Costa Rica of a firefly larva that's eating a gastropod. You can go and watch the full video there. But this vertebral proboscis is a characterization of fireflies. So that glowing, gorgeous glow of the summer that you may associate, it's fueled by eating gastropods, by eating escargot. Here's some look at the front end. This is the last thing that many of those snails would ever live to see. A couple of final words about snails, about their reproduction. They can be gonochoric. They're largely hermaphrodites. They have a trochophore larva that's often or usually suppressed, so they just have this villager larva with a lot of diversity in parental investment. There's yolk for eggs. There can be protein, egg cases, and brooding. Just as you might expect when there's a high correlation between there is a large diversity of snails and there's a large diversity in how they invest in parental.
Now, in midair, they move
And
to
if
the
you're
next
interested,
stage
you
in
can
their
go
pairing.
and see some gorgeous videos from life in the
Each
undergrowth
everts its male
about
organ from
intertwined
just
mating
behind its
snails.
head.
And if that turns your crank, I suggest if you haven't already seen it, you go and watch micro